Hi, my name is Christopher Height, and I write the blog Tom Paine's Ghost. I've been writing it for about 5 years, and I've suddenly been imbued with a new energy for blogging, and specifically video blogging, as I see no reason why not to record in the most full way possible what I'm thinking at any given moment. So I have this subscription to Nature magazine that I got last year, and I got this little insert with it. It's kind of a big like promo ad for RNA purification technologies by this Zymo Research, which I've used their products. It's a good company, but this came as like an addendum with one of the Nature magazines, and what it is the new RNA world. And I've been reading it tonight, and I just started as a postdoc in the biology department at Emory University. That being said, my thoughts and my opinions are my own and do not reflect those of my lab or the university. But I just wanted to start to put my thoughts out there as I have them so that I can get as immediate feedback as possible from anybody who's potentially interested in this. Now, a couple of days ago, I posted a question on ResearchGate that basically asked if anyone out there has any indication of evidence that RNA, small RNA molecules, go with chromatin through replication, meaning, so we have cells that have genetic information inside of them and stored in chromosomes that are loosely packed as chromatin in the interphase part of the cell's existence. But then as cells divide, they replicate all their DNA, and then they come together, the chromosomes come together, and then they align in the metaphase plate, and then so there's interphase, and then prophase, then metaphase, then they start to separate apart, then anaphase, then telophase, and they split apart. Now, my question is, here, during the condensation of chromosomes during mitosis, is it possible that you have these nucleoprotein complexes that not only are DNA and chromatin architectural proteins, but also DNA, chromatin architecturals, and non-covalently attached RNAs, such that the RNAs can tether and kind of piggyback through mitosis, and therefore potentially have downstream effects, or I don't know if I want to say downstream, but effects later on in the two daughter cells' existence. So I learned from a graduate student who's also in my lab about this phenomenon of paramutation. So paramutation, in very simple terms, is when a gene for a trait, or a gene for a phenotype, so let's just say there's a gene for the color red in corn. Okay, so paramutation happens, as I understand it, correct me if I'm wrong, when a gene for, say, the color red continues to express as a phenotype even after the gene for that red phenotype is not there anymore. So it's kind of like a phenotypic echo through some generations. Now, we talk about epigenetics and epigenetic effects, and it seems like a lot of these epigenetic effects are just like that. They're echoes in the germline of whatever species. So Jad Amarad and Robert Krulwich, they produce a podcast called Radiolab, which is one of the most phenomenal artistic scientific marriages that I've ever witnessed. And they did an episode, Carl Zimmer was interviewed on it a few months ago around Thanksgiving time, called Inheritance. And in that they talk about these Swedish studies in which the church records back in the Middle Ages are so precise that they can tell when a kid in Sweden in the Middle Ages would be prone to heart disease and lessened lifespan based on the diet his grandfather had. The principle being that if the Swedish boy in the Middle Ages' grandfather 2 generations back was obese or well-fed, that they would have a higher rate of heart rate and a lower life expectancy. But if their grandfather was starved during the formative years of the germ cells, so between ages 9 and 12, then that boy would have a longer lifespan, lower rate of heart disease. Now, there's a tendency in science now to accept that post-translational modifications to histone proteins, which are the proteins that kind of package the chromosomes up and help hold together all the DNA, that modifications to those proteins somehow get passed down. They're still not really clear on the mechanisms, and that epigenetic, so epigenetic, the word epigenetic just means above and on top of genetics. And so methylation of cytosines, acetylation of various things, modifications that are not at the core of the genetic code, meaning they're not part of ATGC, are passed down somehow. Well, I'm proposing, I have a different hypothesis about how epigenetics works, and I'd like to hear your thoughts. And if you know anybody that's interested in this, who researches this, please pass this along, because I'm really curious about this. I'm really curious if there's any evidence out there that shows that RNA can piggyback through cell division after the DNA has replicated, and then have epigenetic effects down the line. Because if that is the case, we should be able to detect it through like fluorescence in situ hybridization, but with RNA probes that are there for some persistent gene that is still having a phenotype, even though the primary gene isn't there anymore. I'm thinking about experiments that could be done to either prove or disprove my hypothesis about the piggybacking RNAs. Now, there are RNAs, or pi-RNAs, which are Pee-wee interacting RNAs. There are mRNAs, which are just regular messenger RNAs. There are microRNAs. There are small interfering RNAs. There's turning out to be this huge swath of different RNAs, a gamut of various length RNAs, and that's what this Peanuts New RNA World insert is talking about, and it goes through the timeline of the discovery of all those various different RNAs. So, as someone who studied a gene, or a protein rather, well, the gene product, methyl CpG binding protein for my PhD, and I also used MECP2 and did RNA binding experiments, though I never published those experiments. There have been RNA binding experiments with MECP2 published before. I am wondering if there aren't a whole slew of chromatin architectural proteins that, yes, bind DNA, but simultaneously bind and carry with them RNAs that then have either protective or iterative or carryover effects on different cell lineages. And maybe even this is an explanation of cell differentiation and determination. But anyway, food for thought. These are my thoughts tonight, and I'm just wondering if there's any discussions out there. If you have a response, please post it here to YouTube, or just write me an email, christopherhite@gmail.com. Okay, have a good night.